So if there are no further questions, I think we can move on to the next presentation, and that will be on development of nanoparticles for clinical use and the importance of degradation and excretion by Skotland Center for Cancer Biomedicine, the Norwegian Radium Hospital, Oslo, University Hospital, and University of Oslo, Norway.
So, thank you. What I'm going to tell you is mainly based upon my experience of heading work with biodistribution, metabolism, and excretion for all type of contrast agents in one of the main pharmaceutical companies in the field. It was the Oslo-based Nycomed that was bought by Amersham in 1997, and Amersham was bought by GE Healthcare in 2002. And due to a downsizing there, I decided to leave the company, and I'm now working with basic research in the main cancer hospital in Oslo. But I had then experience of bringing 5 products to the market and another 5 into clinical trials. So the topics I'm speaking about are mainly focused upon that it's very important that we improve the quality of biological studies in order to bring nanoparticles further to the market and into clinical use. And in my opinion, the key there is we need more cross-functional collaboration with real experts in the different fields. I'm also commenting upon that researchers in the field that would like the products to get to the market, they should be aware of that there are very large differences in the studies needed and the risk and cost of development of different type of nanoparticles. And that is discussed in detail in this article. I will shortly touch upon some of the issues. So, what is important for part of this discussion is that when nanoparticles are intravenously injected, they have a very restricted ability to escape blood. They can pass through some pores or fenestra in liver, spleen, bone, and some smaller ones in kidney. And they can also pass out through more leaky endothelium in inflammation areas and in solid tumors. And very small nanoparticles can be excreted through kidneys. When discussing which nanoparticles size that can be excreted in kidneys, I think this article from 2007 is a very good one. And it illustrates how the different sizes, hydrodynamic diameters, have effect on how, to which extent the particles are excreted in urine or not. These data are very often referred to as nanoparticles of 5 nanometers are rapidly excreted in urine. I don't agree with that statement. I think it's much better to say that nanoparticles of approximately 5 nanometers, you can expect approximately half of them to be rapidly excreted in urine. But for nanoparticles larger than 10 nanometers, you cannot expect any of them almost to be excreted. And due to this, it's so important that the nanoparticles are degraded in order to be excreted. And this is very important both for safety issues and for regulatory issues. Of course, this may not be a problem for endogenous substances, for example, liposomes made of phospholipids or albumin-based nanoparticles. And it has been shown for the iron oxide particles used for MRI for more than 20 years that they are degraded and excreted. But for other nanoparticles, there are very, very little degradation data available. So regarding targeting of nanoparticles, we have, of course, the passive targeting taken up into liver, milk, and so on, spleen, I mean, and you have the enhanced permeability and retention effect, the so-called EPR effect. When it comes to active targeting, it's important to remember that they are not actively seeking the target. They are just floating around, and there is a selective retention. I think, in fact, that selective retention would be a much better terminology than active targeting. A key question is then, how much of the injected dose can you expect to end up in the target? Most often you see data similar to this, that you can increase the amount taken up in the target by a factor of 2, 3, 4 by having a targeting principle. But this question is, how much is in the target and how much is in the rest of the body? That is very important. What is in the rest of the body is extremely important regarding all safety issues. And if you speak about a contrast agent for medical imaging, well, it's important how much that ends up in the target, but it is even more important what's the ratio between what you see in the target and the surroundings. And then it's very important how fast the agent from the surrounding of the diseased tissue is excreted. In fact, the only data I have been able to find for quantification of how much nanoparticles that can end up in diseased areas in humans is this publication from 2001. They used pegylated liposomes, something similar to doxyl calyx, and have added radioactive labeled substance in it. And they conclude that in this case, in human tumors, 0.5 to 3.5% of the injected dose end up in the tumor. That's the only paper I have been able to find about this data. So, also as mentioned by a previous speaker in this session, there is very little of the injected dose of a targeting molecule that can be expected to bind to the tumor. There are a lot of such data even 20, 30 years ago, when people aim to use, for example, monoclonal antibodies or other targeting molecules for medical imaging and radiotherapy. But it's also important to remember that even if this number is very small, it can still be very important to have this extra effect of a targeting molecule, for example, in a nanoparticle homing into an EPR, because the increased amount of drug that you can get into the cell can be very important for the biological effect, although it's quantitatively not very high. And finally, I would like to stress an important issue when you read literature about how large is the effect of adding a targeting principle, and how much do you increase the binding to the target. And in fact, it's very seldom that you in such studies see the necessary control experiments, so that you can be sure that what you observe of an increased binding to the target is really due to a targeting effect, and not only a change in charge, for example. Some comments about documentation needed for market approval of drugs. And you need then to document all different compounds in the product, impurities, excipients and everything, and you need to document that you are able to make the product in a reproducible way. And if you then increase the complexity of the product, you not only increase your workload, what is needed to document that you are able to make it reproducible, but you also increase the risk that you will not be able to make it reproducible. Regarding safety studies, even for a contrast agent that is expected just to be injected once, you need to perform studies in 2 species, normally rats and dogs, 3 different doses, and with repeated injections for up to 28 days. So if you then have a nanoparticle which is not excreted, then you will need to perform such studies for a very long time. Increased risk, increased cost. There exist guidelines about this, how long time you need to perform such studies, but you need to remember that they are nothing more than guidelines. You have to be discussed in every case. I will not go into detail about specific toxicity studies, but some few words about the area I have been working most with, the ADME studies. And of course, when you then increase the complexity and have more compounds in your product, you have more analytes to look for. These need to be performed according to GLP if there are safety studies. And one question I am really wondering about, what will the regulatory authorities ask for the documentation for such products? Once again, guidelines exist, but they are nothing more than guidelines. Some few words about cost of development and studies. Some numbers, estimates for imaging agents and therapeutics. To my knowledge, there are no such data available for nanoparticles. What can be said is certainly that more constituents make more analysis, higher cost. You can expect higher cost for making nanoparticles based on non-endogenous than for endogenous substances, and they will be higher if they have nanoparticles that are not degradable compared to having them biodegradable. Some few words also about if you are in a situation that you would like to contact the pharmaceutical company in order to ask them to develop your product and to market it. You can expect that they will perform a lot of different type of evaluations, and I will not go into this in detail. You can expect that such questions will depend from company to company, and it will also include that what do the company expect will be the market situation when you come with your product. If you are successful and come with your product to the market, what will they expect to be the situation then? Finally, some few words about theranostics, products that can be used both for imaging and therapy. No doubt that they are very useful for basic studies to understand mechanisms in cells and in animals, but when it comes to humans, I am not that sure. That is partly due to that you make much more complicated product. The product need to be approved for each separate use, and they are probably not the same optimal properties for therapeutics. They should probably circulate for a longer time than for an imaging agent. So I think it's better to focus on agent for one specific use. And if I am going to try to summarize from my long experience in pharmaceutical industry in one single sentence, it sounds like make products as simple as possible. Finally, just some few words about that. It's very important that we have cross-functional collaborations between experts in different fields. I will not list all this, just state that we have taken the consequence of this in Norway by building up a national competence building project involving all these type of expertise with 10 different research groups, and we have a poster just outside here describing that project. So finally, I would like to acknowledge the contribution for people in the group, the head of the group, Professor Kirsten Sandvik, and also Tore Geir Iversen, and thank you for your attention, and mention that we have a research position available in a cross-functional project involving people in Belgium and India, and to start in October this year. Thank you.
Nice presentation. Now the questions can be asked during this next session that we are going to have for half an hour. All the speakers are requested to come up on the podium, and I think the audience and the speakers, and also between the speakers there can be interaction. So I have some question, but I will ask afterwards.